Hello, everyone, and welcome to today's webinar, Introducing REM I Platform, a benchtop instrument, AI model, and data suite for single-cell high-dimensional morphology analysis. I'm Michelle Balakrishnan, a Senior Product Marketing Manager at Deepcell, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Deepcell. To learn more, visit deepcell.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues there as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Courtney Anderson, Director of Product Marketing, and Ryan Carelli, Senior Data Scientist, both from Deepcell. Courtney, the floor is all yours.
Hello, everyone. Thank you for joining us today. My name is Courtney Anderson, Director of Product Marketing at Deepcell, and it is my great pleasure to introduce to you REM I, our new platform which takes the power of morphology and magnifies it with AI. It is well known that cell morphology can be highly indicative of a cell's phenotype and function. For instance, here we can see a homeostatic microglia whose morphology indicates a healthy environment and surveillance state. However, reactive microglia respond to a trigger and release pro-inflammatory cytokines, proliferating and changing their morphology dramatically. And if there is neuronal degeneration, microglia will further alter their morphology and engage in phagocytosis. Now, these differences are very clear and striking to the human eye. But in this case, not so much. Here are two cells, one that is drug sensitive and one that is drug resistant. By eye, it is very difficult to tell them apart. Cell morphology is highly dynamic and complex, and analyzing morphology by eye can only tell us so much. And so this demonstrates the strong need for a more quantitative, high-dimensional, unbounded approach to assessing cell morphology. A more quantitative, high-dimensional, unbounded approach would magnify the insights into a cell's phenotype and function and turn cell morphology into a quantitative, high-dimensional ohm, or the morpholome, similar to how methods have been developed to assess the genome, transcriptome, and proteome at high dimensions. The problem is that current methodologies to assess and characterize cell morphology have been limited to either imaging or sorting with labels. Until now. At Deepcell, our approach to morpholomics is to provide single-cell, high-dimensional, unbounded morphology analysis. We offer reliable, rapid, and standardized collection of bright field images of each cell in a sample. We have foundation deep learning models for high-dimensional feature extraction of known and novel cell characteristics. We incorporate label-free sorting of morphologically similar cell groups to gain further molecular and functional insights. And lastly, we offer interactive software to store, visualize, and analyze cell images and high-dimensional cell descriptions. And today, I would like to introduce to you the REM I platform, which provides high-resolution imaging of single cells, high-dimensional analysis of morphology, and label-free sorting all in one platform. REM I directly takes real, high-resolution, bright field images of each individual cell and is powered by Deepcell's human foundation model for high dimensionality. REM I also has the ability to sort cells 6 ways based on their morphology without the need for any labels. Furthermore, you can collect these viable, minimally perturbed cells for downstream analysis, such as single-cell RNA sequencing. And it is an end-to-end solution with our Axon data suite, which enables real-time data visualization and analysis from cell images obtained on the REM I instrument. So let's dive into each of these components a bit more. First, I'd like to highlight the REM I instrument. This is an AI-powered benchtop instrument for high-dimensional morphology analysis. Some of the key features of this instrument is that it takes high-resolution, single-cell, bright field images. This is also a completely label-free workflow, and we can also sort 6 ways. By doing this, we can maintain viability of the cells for downstream analysis. And as I mentioned, the AI component means that there's real-time characterization of the cells through AI using our human foundation model, which I'll talk about in a moment. We also offer flexible experimental designs and a fast workflow on the instrument. Now here's a depiction of the instrument and some of the key components. We offer two types of kits. The REM I imaging kit, which consists of reagents and consumables, including our imaging chips, for those interested in doing an imaging-only workflow. If you'd also like to sort, we have the REM I sorting kit. This consists of reagents and consumables, including our sorting chips, for those that would like to do the imaging plus sorting workflow. And you can see here a depiction of the instrument with the drawers open. We have the reagents drawer where all of the reagents that Deepcell provides will go. And then under the main door, you see where the stage for the chip loading is, the sample tube where your sample preparation would go, and then the negative outlet tube, which is where it collects all of the cells that do not get sorted. One of the key components of the REM I platform is the Deepcell Human Foundation Model, or HFM, which assesses 115 cell morphology dimensions. It combines deep learning and computer vision methods to extract cell features that quantitatively and reproducibly represent cell morphology. The deep learning aspect of the HFM is based on a self-supervised learning algorithm. It's trained using millions of cell images from many biological human samples, and it detects differences in cell morphology without labeled training data, meaning that the HFM is ready to use on day one without the need for additional training. The Deepcell HFM is unique in that it combines deep learning and morphometric features, providing both accuracy and interpretability, and it is a fast and effective cell feature extractor, enabling real-time cell classification and sorting. The HFM contains 115 combined embedding dimensions, including 64 deep learning derived embeddings and 51 computer vision derived morphometrics. So let's talk about the distinction between these two a bit more. Morphometric features can be considered human interpretable, quantitative metrics of cell morphology, including cell size, shape, texture, and intensity, as well as biological features such as blebbing and pigmentation. They also map to measurable and recognizable concepts. And shown here are some of the examples of the 51 morphometric features that we compute, starting with simple and obvious ones, such as area and perimeter, to the ones that measure granulations and more subtle features. You can find a complete list and description of these metrics on our website in our HFM panel sheet. Deep learning features are information-rich metrics of cell morphology with powerful discriminative capabilities, but they may not be human interpretable. They are quantitative descriptions of cell features derived from deep learning models based on neural networks. And of note, AI-derived features can outperform traditional morphometrics in cell classification tasks through identification on non-human interpretable cell features. Hence why we have combined deep learning with morphometrics to provide both accuracy and interpretability in a fast and effective cell feature extractor. Now, the final component of the platform is our Axon data suite. This enables real-time exploration of data obtained from the REM I instrument. Some of the key features of this data suite is this is where you can store, review, and organize all of your runs. You can also visualize all of those embeddings that I just mentioned as morphology UMaps. You can also view or overlay cell images directly on the morphology UMap. And you can explore these AI embeddings interactively through comparative analytics and graphs. You can also train custom classification models and define cell populations of interest for collection in up to 6 wells, as I mentioned earlier, since this can sort up to 6 ways. And we also have a number of data sets available for you to explore on your own. We have one that shows the identification of cancer cells, exploration of cancer cell subpopulations in a melanoma sample. You can also get immune cell diversity in the tumor microenvironment. And we have the most recent data set we just released where you can explore cell death pathways in Jurkat cells. So please visit exploredata.deepcell.com to look at more of these data sets on your own. Now that I've introduced the three components, I'd like to talk a little bit more about how they come together in a simple and streamlined workflow, starting with dissociation of cells into a single cell suspension, followed by imaging of those single cells in flow, and then characterization of those cells in real time with our AI models, all of which is done on the REM I instrument. And then in the Axon data suite, you can analyze the data. In addition, in this data suite, you can select cell populations of interest in real time and then collect those cells for further downstream analyses, such as single-cell RNA sequencing. You can go from sample to a publishable figure in just a matter of a few hours. Because the sorting process is gentle and label-free, that also means that the cells are minimally perturbed and still viable at the end. This then makes them ideal to use in downstream analyses. Therefore, we assess the effect of the REM I sorting on single-cell RNA sequencing. PBMCs flowed through the REM I platform were compared with control cells stored in medium using either a single-cell targeted immune panel or single-cell whole transcriptome workflow. On the left is a UMAP of gene expression profiles from the pre-sorted and sorted cells, while on the right is a correlation plot for the two conditions. The two samples overlapped with each other in the UMAP plot, and gene expression levels showed high correlations, indicating no significant effect on gene expression after sorting on REM I. We also compared the effects of REM I on cells to that of traditional FACS. Here, human neutrophils were first isolated from whole blood and then split into three conditions: unperturbed, stained and flow sorted by FACS, and unstained and unlabeled and sorted by REM I. These cells were then lysed for bulk gene expression profiling by RNA sequencing. We can see that the REM I sorted cells had minimal gene expression differences compared to unperturbed cells, and much fewer gene expression differences compared to FACS sorted cells. This data demonstrates that FACS significantly induced up and down regulation of pathways in neutrophils, while the REM I platform minimally perturbed the cells. We are seeing the REM I platform being used in a number of key research areas, including cancer research to assess tumor heterogeneity and malignant cell detection and enrichment, in developmental biology for the detection of induced pluripotent stem cells, cell activation and differentiation, and cell cycle states. And also in cell and gene therapy for compound screening, functional screening, and manufacturing QC. And there's many more areas where the technology is being applied. These areas can be bucketed into two broad categories: sample level profiling, which some examples include heterogeneous sample evaluation and characterization, disease detection and enrichment, and drug and CRISPR perturbation screening, and also the cell level profiling, including cell health status, cell state characterization, morphology as a biomarker, and integration with multiomics. And I'm now going to turn the talk over to my colleague, Ryan, to highlight a few examples from each of these categories.
Thank you, Courtney. In this section, we'll show REM I datasets, which are focused on various cell and sample profiling applications. Let's first look at a simple dataset, which is comprised of polystyrene beads, two cancer cell lines, the A375 melanoma and CALB3 ovarian cancer cell lines, and an immune cell line, the Jurkat T lymphoblast cell line. Each class has distinct morphological features, with the highest visual similarity among the cancer cell line images. Images were mixed in silico, with representative images from each class shown in this bottom panel. Then the deep learning and morphometric features were extracted using our human foundation model, as Courtney's described. The extracted features were standardized and projected into a two-dimensional UMAP basis. This morphology UMAP shows a clear separation between the polystyrene beads and cell line images. Among cell lines, while the Jurkat immune cell population is clearly distinct from the cancer cell lines, the two cancer cell lines show greater overlap, which is in agreement with our expectations. When we train a classifier to distinguish these four classes, we see that HFM features that distinguish beads and the three cell lines work with very high accuracy. Class confusion was highest between the A375 and CalV3 cells, which is as expected, and a combination of features contributed to classification. We've plotted a subset of them here. Some are our morphometric features, computer vision features, and others are deep learning features. While morphometric features like the maximum radius and the small set of connected bright pixels distinguish the Jurkat and bead cell images from the cancer cell images, these features were nearly identical between CalV3 and the A375 cell images. But the deep learning features, on the other hand, are able to distinguish the cancer cell lines. For example, this deep learning feature number 11, which is shown in the rightmost panel, distinguishes A375 in green from CalV3 in red. Next, I'll show how the REM I platform can be used to characterize heterogeneous samples. In particular, here we profile the microenvironment of melanoma tumor samples. To establish ground truth cell identities, we also imaged human melanoma cell lines, which represent each of the microenvironment constituents. Then combine those images in silico with the tissue samples. On the left, we see a UMAP, which is based on morphology features and colored by cell type. On the right is the same UMAP, but colored instead by cluster. Using only morphology, we're able to separate the tumor cells from immune and stromal populations in a completely label-free manner. By looking at representative images from each cluster of the immune cells, we can see that immune cells have subtle but separable morphologies, which include variation in size and in granularity. Another notable finding is that pigmented melanoma cells, shown in green to be melanoma on the left, and represented by these cell images, are morphologically distinct and cluster separately from non-tumor cells. This is unique as current technologies, such as flow cytometry, will require known markers to detect pigmented cells, while with the Deepcell platform, we've detected these pigmented cells based on morphology alone, without the need for labels. If we select only the melanoma cells from this dataset and project them into a new UMAP, we can see that there are three major melanoma groups: the dissociated melanoma biopsies, the melanoma cells from the cell lines SKML 1 and 2, and pigmented melanoma cells. Within this pigmented melanoma group, we can identify two different clusters. By sampling representative images from each of these populations, including non-pigmented and the two different pigmented melanoma cell populations, we see that, especially among the pigmented cells, while both are pigmented, one has blebbing around the cell membrane, which suggests that this pigmented population could be undergoing apoptosis. If we examine the distribution of mean pixel intensities in each cell image, we can confirm that indeed these clusters, 6 and 9, are more pigmented than the non-pigmented cluster 2. In summary, here the REM I platform was able to reveal heterogeneous morphology among these cancer cells, including pigmentation, which can be difficult to identify with conventional methods. All of the data shown here was analyzed using our Axon data analysis suite, so there's no need for additional computational tools or programming to reproduce this analysis. Furthermore, if you wanted to collect these cells for additional downstream analysis, such as single-cell RNA-seq, you can easily select the cell population of interest in the software, and the instrument will sort the cells for you, keeping them viable and intact. This dataset is available to explore for yourself on our website. It can be found at this URL, exploredata.deepcell.com. In the next dataset, I'll show how REM I imaging can identify characteristic morphologies of cell health states. Jurkat T lymphocyte cells were treated with cytotoxic methods, camptothecin, staurosporine, and heat. Following this induction or treatment, the cell death was quantified by flow cytometry, using annexin 5 as a marker of apoptosis and cytox co-expression as a marker of necrosis. By imaging each condition, we can then study morphology as a readout of apoptosis or necrosis. In this top row, we have flow cytometry plots containing estimated fractions of viable, apoptosis, necrosis, and dead cells. We've organized the cell health state roughly left to right. In the middle row are the corresponding morphology UMAPs. These are based on REM I images of cells from each condition, and the color is according to the proportion of cells that are in each region of the UMAP plot. So cells are accumulating in the area of the plot which is more red. You can see that cell morphology is clearly altered between conditions. And by looking at cells sampled from each condition, you see that each condition has characteristic morphological features. Notably, cells which occupy the same flow cytometry gate can be distinguished based on morphological features, which indicates that morphology can monitor subtle changes in sample health status and assess sample quality. For example, here we show 18-hour 2 micromolar staurosporine treatment next to a 5-minute 70C heat shock, which by flow cytometry have similar necrotic proportions. But staurosporine treatment is known to represent a more advanced stage of necrosis. And this difference can be detected with REM I imaging. In this next dataset, we'll show how REM I imaging can distinguish cell lineage, type, and state in PBMCs. For this dataset, we've included B cells, CD4 T cells, NK cells, activated CD4 T cells, monocytes, and macrophages, which have been purified from multiple donors. These purified samples provide a ground truth reference against which we can compare images of unpurified PBMCs from 3 donors. The first comparison we'll make is at the top of this hierarchy between myeloid and lymphoid lineages. Lymphoid and myeloid cells are expected to be distinguishable. Morphological differences have been documented in the literature. They include prevalence of vacuoles, differences in the prevalence of vacuoles, distinctive cytoplasm, and distinctive nucleus. In addition, simple features like size and granularity, which are often measured using the FACS forward and side scatter, can be used to isolate these lineages. When we apply our human foundation model and fit a classifier to identify the morphology features that differ most between the myeloid and lymphoid cells, we find measurements of size and shape, maximum radius, maximum caliper distance, long ellipse axis as expected. In addition, we find deep learning features that are even more discriminative, as indicated by the high importance scores, and also the clear differences in the distributions of these features in each class. This classifier achieves an accuracy which is greater than 90% in distinguishing lymphoid and myeloid cells by morphology alone. Next, we'll look at how REM I imaging can distinguish differentiated macrophages from monocytes. These classes are again separated with high accuracy, greater than 90% by morphology alone, and we find that deep learning features are top differentiators, which emphasizes the value of deep learning to this technology. In this next dataset, we'll show an exciting study that we did, monitoring drug response using morphology as a readout. We treated K562 lymphoblast cells, which are isolated from a leukemia patient, with chemotherapy drugs. We were interested to see how morphology changed over time with treatment, and we're especially interested in morphologies which might be associated with resistance. Shown here are the results of a time course experiment where these cells were imaged 12 hours, 3 days, 5 days, and 8 days after treatment. We collected a separate time series from controls in treatment with two chemotherapy drugs, a hypomethylating agent and a histone methyltransferase inhibitor. Each of these drugs resulted in cell death, which can be seen by the movement of these cells to the far right compared to the controls. But what's exciting is that at 3 and 5 days, pockets of cells which were resistant to treatment could be identified by morphology and are isolated for further study with other assays. In this final data set, we'll show an example of morphology as a biomarker. Morphology can be used as the basis for population analysis of a heterogeneous tumor sample, in much the same way that we commonly use biomarkers. Here's the workflow. Using morphology analysis, multiple morphologically distinct cell groups can be identified within both the biomarker positive and the biomarker negative tumor fractions. Now, these morphological groups may correspond well with the biomarker, or in many cases, they'll offer additional information on top of the biomarker. With REM I, we can sort each of these morphology-based cell groups. Then the cells can be subjected to molecular assays to assess the functional significance of these cell groups. In this experiment, we used CRISPR to knock out 11 genes in human embryonic kidney cells. These genes were previously linked to morphology, and impacted morphological features included cell size, nuclear size, and vesicle formation. In the morphology UMAP on the left, we see 4 distinct cell morphologies which were induced by the knockout. Each of these morphologies was then sorted and processed by single-cell RNA-seq, which produces the transcriptional UMAPs that we can see on the right. Notice that the transcriptional profiles are shifted, but not nearly to the extent that morphology changed, which indicates the advantage of morphology in studying certain biological systems. Although the morphology groups don't separate clearly by single-cell RNA-seq, they do have significant transcriptional differences and signatures, which we can use to link morphological with molecular differences. I hope that these data stories have convinced you that the power of morphology can be magnified by AI with Deepcell's REM I platform, which is a fully integrated solution for single-cell, high-dimensional, and unbounded morphology analysis. I'd finally like to thank our many collaborators around the globe, as well as all of you for joining us today. And now I'll pass it back to Michelle.
Perfect. Thanks so much, Courtney and Ryan, for the lovely presentation. We'll now start our live Q&A portion of the webinar. If you have a question, please go ahead and submit it in the question box on the left-hand side of your screen. Alrighty, pulling up the questions here. First one for you, Courtney. What is the viability of the cells you get out of the system?
Okay. Yes, thanks, Michelle. That's a great question. So really with this system, you will get greater than 95% of the input viability. So that means that, essentially the viability of the cells collected will be at least 95% of the viability that was put into the run. So if you had 100% of the cells viable at the start, at least 95% of those cells will be viable at the end of the assay. And this is actually based on a wide range of cell types that we have tested.
Thank you. Also for you, Courtney, kind of general question. What kind of samples can you run on the REM I?
Okay. Yes, that's also one I'd like to really clarify. So with this, really the samples that work best are dissociated human cells. They can be either cell lines or primary cells that are taken from human tissue. And ideally, they should be within 6 to 25 microns. Those really work best for the cell types. But we have tested a number of sample prep types with the platform. And this includes cryopreserved cell lines that are either in suspension or adherent cells after you trypsinize the cells. We've also tested, obviously, cells from dissociated tissues, such as dissociated tumor cells, and also fixed cells, including PFA fixation. And one other note, too, is to think about the species. So our model, the HFM, or Human Foundation Model, has been trained and tested on human cells. However, we have seen success in applying the model to other mammalian species. So I want to give some clarification there on the sample types.
Perfect. Thank you. Ryan, got a question for you. How does the software define what a cluster is?
Yeah, that's a great question. For every cell, we're extracting features using our Human Foundation Model. These features are designed to represent cell morphology, so they can be used to determine morphological similarity or differences between cells. We call a group of cells a cluster if all of the cells within that group have similar morphologies to each other, but different morphologies to cells outside of the group. In our Axon data suite, we've chosen to use a modularity-based clustering algorithm called the Leiden algorithm, which is very popular in single-cell analysis. But any clustering method could be applied to the data.
Thanks so much. A common question that we get, I see today is no different. Can you use fluorescence on the instrument?
Sure, I think I can take that question. So the instrument captures bright field images, not fluorescent images. But I really think that's the beauty of this platform. It's entirely label free, so there's no need to fluorescently label your cells or use fluorescent tags. So this really enables unbounded hypothesis generation and true discovery applications with this method.
Perfect. And I think you touched on this a little bit in your sample type answer, but what are the cell size limitations? Probably we can reiterate that.
Yep, that's another really good question. Yes, as I did mention earlier, the 6 to 25 micron diameter does work best, but we do have evidence to suggest that larger and smaller cells could actually also be successful with the platform. We've run cardiomyocytes successfully, which basically an example of a large cell type, so something larger than 25 microns. And we also have evidence with smaller cells, such as T cells and B cells. They've been able to work successfully with the platform. So going down to as low as 4 microns and potentially up to 30 or slightly more on the larger sides of the scale. But I would say the standard application input cell size is 6 to 25 microns.
Do you have any data with neurons that are more fragile for standard flow cytometry? Examples looking at neuron differentiation.
I can start this off, and then Ryan, if you have anything to add into this. I would say that we are exploring this right now. We really have seen really great applications within the cancer and immunology space, and neuroscience is definitely another key area that we are very much interested in exploring with this assay.
Okay. Then we've got, I'm not sure if this question is specifically about our chips or whether it's about using tissue slides, but do you use your custom slides or any other form of commercially available are allowed?
Mm. Yes, maybe to clarify, so this doesn't use any type of tissue sections or slides or tissues that are on slides. But as Michelle mentioned, this does utilize chips. So we have the imaging and sorting chips, as I had mentioned earlier. These are basically chips that would be loaded into the instrument, and then these cells flow through them. And if you're using the sorting chip, that's where you would end up collecting your cells on there. And this is something that we provide to you as part of the reagents and consumables. That all comes from Deepcell.
Another question for you, Ryan. How do you obtain those deep learning features?
Yeah, so we've trained a model which uses a method called self-supervised learning. So this is a method where we take a very large data set of cell images, and we use those cell images to extract sort of features that describe all of the morphologies that are present in the cell images. So this basically requires no labels and allows us to sort of expose the model to a huge variety of cell classes, enabling the model to generalize across a very wide variety of cell types, organ types, and so on.
Have we tried native leukocytes in plasma or only isolated into cell culture media?
Sorry, Courtney, do you know the answer to this? I don't know the answer to this one.
I will say I'm not aware of being able to try this sample type, but that's something that we can consider. So.
Okay. When using the sorting function, can you collect single cells within a given population?
Yes, that's a great question. So, with the sorting feature, you would collect the cells that are from that same morphologically similar group that Ryan was discussing, but you cannot pick out each individual single cell from that population. You would be sorting for that homogeneous, morphologically similar population.
Okay. And Ryan, for you, based on the deep learning model, uh, so it should be fit for every cell. Could it be applied to differentiate cells in a different status?
Yeah, something that distinguishes us from other deep learning based imaging platforms is that we don't need to fit a new model for every cell type. Because of this self-supervised learning method that we've used, we're able to collect a diverse, very large pre-training data set and extract all of the features from that data set so they can be applied to samples without you having to train new models. I want to point out also that we don't only use the deep learning features, we also use these morphometric features. So combined, we found that the models generalize very well to a variety of samples, human samples.
Perfect. And kind of an elaboration on that, there's a question on how many cells are in the database. I'm guessing that's referring to the training data set. And how many cells do we define as a critical mass for the software?
So I can take the
So,
how many cells are in the
yeah.
database, which is many millions, and we're constantly expanding that database to iterate new and better models. Maybe the critical mass is about the cell requirements for running the instrument. Courtney, do you know the specs for that?
Yes. So I would say that in terms of the number of cells that can be run on the instrument, if that's what this is question is referring to, we do have a pretty high input cell count that is basically could be up to anywhere from really millions of cells, depending on whether they're going to be live or fixed. So it really does matter. We can go up to 3 million, 10 million. We can go, as low as the lower end of the cell input is needed.
And Ryan, following up, in case this question is getting at something else, like to train a classifier, is there a certain number of cells you need to have already run through in order to train that classifier?
Sure. That does depend a little bit on the complexity of your sample, but on the order of hundreds of cells is the minimum from a modeling perspective.
Thank you. Okay, it looks like the number of questions are starting to wind down. So I'll just go ahead and say thank you again to Courtney and Ryan for your time today, for this really awesome presentation you gave. Also like to thank Labroots for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today, for your great, interesting questions. Any questions you might have that didn't get addressed or come through the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's ready for replay. And we encourage you to share that email with any colleagues who might have missed today's event and might be interested in this type of research. Until next time, take care and thanks so much. Bye now.